Thank you. Everybody can hear me? Great. Okay, well, my name is Max. I am the head of computational biology and data science at Tally Health. And today it's my privilege to give you an overview, a little bit of our company, and also give you kind of the scientific details behind our next generation buccal clock that we published earlier this year. So Tally's mission is to help everyone live healthier longer. We launched in February of 2023, so we're a direct-to-consumer longevity company. We work directly with people. And we help people by providing 4 key products. The first is epigenetic tracking using a unique and painless cheek swab test. We also have personalized lifestyle recommendations that are function of both what you're not doing, what you're doing, and also the epigenetics. We have expert-crafted educational content that we're very proud of. A lot of cutting-edge stuff that's coming out in the literature, and also science-backed supplements that we formulate in-house and have stringent QC. So we've been featured in many exciting articles and journals, but I think my favorite is now ARDD2024. This is a wonderful conference. As Morton said, I was here last year, and I made it my mission to come back, and we had to sponsor because of just how awesome the talks are, how diverse. So it's great to be here. So when we started out, there wasn't a cheek swab clock, a buccal clock, that was publicly available. So we started out without having a clock, and we needed to build one. And so, how do you do this? Especially if, we haven't even launched the company yet. So the approach that we took, we reached out to over 200,000 volunteers on our wait list, and we asked them to provide, basically answer questions about their lifestyle, their health, and their demographics, and consent to take a kind of like a beta test. And we were surprised. We got over 25,000 people to agree to do this, and they were eager to try it out. So from that group of 25,000, we picked 10,000 of the most diverse people that, we wanted to make sure we get healthy people, but we also get people that have maybe less healthy lifestyles. And we sent them a test. We got over 8,000 tests back. And so when we look at this cohort, you can see that it's very diverse. We have people aged 18 to 93, and we've tested it even beyond, up to 100. We see roughly 50% males and females, and we have 33% non-white demographics. In addition, we asked 190 people to do 2 swabs. So we're able to collect biological replicates, which is important for us because as we develop the clock, we want to make sure that it's as reproducible as possible. So just some interesting highlights from the survey information that we got. So we asked people to self-assess their health, how they perceive their aging, their stress levels, their immune health. And so here I'm showing you kind of box plots of the distributions for the age. And as you go from left to right, you're going from the least healthy to the most healthy, or the most stressed out to the least stressed out. And what we see is that there's a bias for people they feel like they're the healthiest and they're aging the best. They tend to be older. And so I think, maybe this is a survivor bias, maybe this is a, generational effect, but I like to think it's because older people tend to be wiser and they tend to be happier. And we know that this has been reported elsewhere. So aging is actually maybe not that bad. When we look at differences between females and males, one of the things that jumps out is BMI. So we see that for higher BMI ranges, females tend to be older than males in our population. But however, as you age, typically BMI decreases, which is in line with what's been published. So we assayed the buccal samples for DNA methylation, and we selected DNA methylation because we know that it changes with age, and there have been many clocks that were built on this technology. And it also changes with lifestyle. But also very important, you can reverse it, right? So you can go back and forth. So we decided to use the Illumina Epic Array for all 8,000 samples. And if you just do a basic principal component analysis, so you just kind of look at it using a reduced number of dimensions. I'm showing you here a heat map that's been clustered, and the colors represent the correlation with a particular variable of interest. So, for example, chronological age, or whether you're smoking or drink a lot of alcohol. And what we see here is that there's very high correlation with specific components in this data set. And importantly, every single one of these is represented somewhere in the top 18 components. So this is great because that means that we just inherently we have information about what we care about in this big data set. As an example, here I'm showing you chronological age plotted as a color. So it's a gradient from orange to purple along the top two principal components. You can see that this is actually a very clear signal that comes up. So the first thing we tried was to build a simple linear model that's just been penalized for complexity. And you can do this, and it gives you a very accurate predictor of chronological age. However, when we looked at the associations of the delta age, or the predicted age minus the chronological age, the difference between the predicted and your chronological, and then you look at the correlation with lifestyle, what you see is you basically have no correlation, right? So if you're just doing this kind of just looking at chronological age, it's not enough. The only one that showed up was alcohol, and it went in the wrong direction, so we're like, no, we can't use this. So we had to go back to the drawing board. And so how do you build a next generation clock without longitudinal data? The approach that we took is called multi-objective optimization. So the idea is to optimize for multiple things at the same time. So here we have a score, and that score is a function of the accuracy, the complexity of the model, which is important for making sure the model generalizes to other data sets. But then we included this green term for the correlation with lifestyle. And this is actually the key here. So when we do this and we say, we really care about the correlation of the delta age with lifestyle, and we add that explicitly into the optimization, what you can see if you use a stochastic optimization approach, if you look at the score, which is the top left plot right there, you can see that as you go from left to right, so you're optimizing, you're taking steps in the optimization process, you see that the score drops right away, so you have an improvement right away, and this is due to this RMSE, or the accuracy, improving dramatically, and this happens very quickly, so it's easy to get a very accurate age predictor. However, if you look at the lifestyle correlation, which is this green trace here, you can see that basically the lifestyle correlation does not improve for a very long time. So you have to actually keep optimizing, and eventually, about halfway through, you start to see improvements with lifestyle correlation as the kind of the training finds a solution that is both accurate and correlates with lifestyle. And if you look at the complexity increases, right? So as you're optimizing, it becomes a more complicated model, but then as we force it to become a simpler model, it's able to find a simpler solution that's still accurate and that still correlates with lifestyle. So this is the approach that we took. We call this model cheek age because we're swapping cheeks. And it's very accurate. You can see that it's in a 10-fold cross-validation. You can see that it's very accurate. It generalizes. And what's important here is that now we have very, very strong correlations of the delta age with lifestyle factors. So BMI, smoking, alcohol, social satisfaction, stress levels, your exercise, sleep quality, your plant-based diet, and self-rated health. And what's interesting here is that social satisfaction and stress levels actually were were higher up in terms of their association with the delta age than something like exercise, which is a little bit surprising. But this is a kind of a feature of our model. So one of the things that we wanted to do was make sure that we improve test-retest reliability as much as we can. This is important if you want to track over time. And so one approach that we found, one trick, is instead of using the methylation inputs directly, the idea was to now average methylation inputs from very similar looking CPGs. So the idea is to first cluster everything and then use the averages of the clustered CPGs as the input. And when you do that, you find that it improves test-retest reliability by about 25%. And again, we're able to do this because we had replicates that were swabs within a 24-hour period. The other thing that actually was a little bit surprising to me, even though actually makes a lot of sense in hindsight, was if you actually use more than one model, like more than one clock. So we ended up, since we're using a stochastic approach for optimization, each time we train the model, we get a different solution. So if we repeat this 1,098 times, which took a long time and cost a lot of cloud computing, but once you do that, you can then rank the models based on their scores. And if you pick the top 100 and use a weighted average of those predictions, you end up improving the reproducibility by another 35%. It also improves the accuracy, and it improves the lifestyle correlation. So ensembling is actually a really, really awesome method for this. Just looking at the clock, it's a pretty complicated clock because it consists of 100 different models. But if you look at the lowest weights and the highest weights, so if you look at the lowest weights, you can see an enrichment for things like the stress-activated MAP kinase cascade. You see platelet-derived growth factor receptor beta signaling come up. And you also see regulation of mitochondrion organization. So this is showing us that there is some sort of biological signal that's being captured by this clock. If you look at the weights that are the most positive, so these are the weights that tend to increase your age, you see a lot of differentiation in development terms. You also see signaling, you see autophagy, you see protein localization. So I think this shows to us, at least it reassures us, that there are relevant biological processes that are being picked up on by this training. So, these are all very relevant to aging, and I think this is pretty cool. Okay, so the next thing we did to validate our clock, first we checked that it works in other buccal data sets. I'm not showing this here, it's in the paper. But we can show that this works in an independent data sets for buccal, and the accuracy is still very high. But we also wanted to see what this looks like in other data sets that have some sort of health variable. So again, these are typically done in blood, so it's a different tissue. Maybe it's not ideal for this particular clock, but what's important is the difference between the conditions, right? So here, I'm showing you again, box plots of comparing COVID positive and other infections to non-infected. So these samples show a significant increase in the delta cheek age. So you tend to be older than your chronological age by a significant amount if you have COVID. We also see a significant association with meningiomas, with HIV retroviral adherence, so treatment adherence. You're actually younger. And we see other associations with progeria, with childhood cancer treatments, BMI, fibroblast passage number, and this is something that was in the paper. We are also seeing associations with pregnancy, so actually pregnancy tends to decrease your age. We see associations with trauma, squamous cell carcinoma, non-alcoholic fatty liver disease, MCI and Alzheimer's, HIV viral load, and major depressive disorder. So even though we're swabbing cheeks, we train this clock on cheeks, we're able to pick up on these signals in other data sets, which I think is really fascinating. So after we published this work, we followed up with another study looking at the association of our clock with mortality. To do this, we reached out to the Lothian birth cohort group from the University of Edinburgh. This is a longitudinal data set that was collected in blood. So they have blood draws over time, and they were assayed for methylation, but using an older array technology. So this is missing half the CpGs. It's in blood. Nevertheless, if you look at the delta cheek age, again, this is the delta between the predicted and the chronological, and then you stratify by the delta cheek age, you can see that for the groups that have the youngest delta cheek age, their 50% survival rate is at 90.9 years in this particular cohort. But then if you compare that to the oldest delta cheek age group, they're more than 7.5 years younger at the 50% survival mark. And this is very significant, but if you just want to look at hazard ratios for every standard deviation of cheek age, you have a 21% increase in the risk of mortality, and this is very significant. It's similar to PhenoAge, which is another next generation clock that was trained in blood and had all the CpGs. So this is reassuring to us because we're actually surprised. We didn't think this might work, but even though it's a different tissue and it's missing half of the inputs, we're able to see very significant associations with mortality. Most importantly, we have tangible benefits for our users that we can now see over time. So we've only been around for about 1.5 years. But one of the things that we can look at is how people are saying they're doing in terms of their lifestyle. So we have a metric called the lifestyle score, which is kind of a composite of the important lifestyle factors you can think of: exercise, diet, sleep, your mental health, and as well as your habits. And so if you combine all of those, the higher the number, the better. 100 is if you're perfect. And what you can see is that over time, as people join Tally, they start out with a pretty good score, but then they increase by over 5 points within the first 3 months, and this is sustained or maybe even improved over time. And just to give you a sense of what that means, 5 points in this scale is like if you didn't exercise, and now you're exercising basically every day of the week. So this is a significant, people are saying that they're being significantly healthier. In addition, if you look at the epigenetic test, so our Tally age test, which is based on the cheek age test, it's inspired by it, the fraction with improved Tally age, as you take more and more tests, increases. So we have about 2/3 of people improving by 6 tests. And if you look at the difference between the last test and the first test, this difference starts out relatively small, even though it's still significant. But as you go up to 6 tests, you see about a 1-year improvement in their delta age after 6 tests. So this is really great because we're now seeing that we're helping people over time. And so with that, I'd like to stop. I think in summary, we have a large, diverse data set, which we use to build and validate an accurate and reproducible next generation buccal clock. It's associated with lifestyle, with health, with mortality. And if you'd like to play around with it, we have a free server that you can upload your own data if you want to predict. You can reach out to me. We can set up a collaboration. I'm always happy to help others out. And if you are interested, we have an exclusive coupon code if you want to try our products out. With that, I'd like to thank my co-authors at Tally. One of them is here today. And the Lothian birth cohort collaborators, which were great. And the beta cohort volunteers, thank you. Without you, we would not have Tally, we would not have the clock. And our funding sources from our investors. Thank you.
Thank you so much, Max. That was really great. Do we have questions? Otherwise, maybe I will start. So with the buccal swabs, you can also do potentially microbiome sequencing and stuff like that. Is this something you are thinking about implementing?
Oh, that's fascinating. I think That is something that would require additional kind of technology development from our perspective. We work with a lab partner, but that is fascinating. Yeah, I mean, we also are thinking about other things you can assay.
10% IP here. 60%.
Oh.
More questions?
Hi, thank you. I'm wondering if you can comment on the pros and cons of grouping prior to analysis based on things like sex or ethnicity.
Yeah, so, I mean, I think it just depends on kind of your distribution of the data that you have. So you could make separate clocks for separate groups, but then you have fewer samples to train on. We found that this is actually the best approach, and it becomes more general, so it's able to pick up on just more general biology if we do it this way.
Hello, thank you. So you said that you had many diseases that had a reduction in longevity, which would explain for the variation between different candidates of the same age. Were these signatures actually so relevant that when accounting for the bigger factors, you could predict a potential number of diseases that the patient would have?
So we don't, I think that's fascinating. It's definitely possible, and I think other clocks have shown something similar. We don't predict disease, but I think it's definitely possible.
All right. Thank you so much, Max. That was really a great talk. Fantastic.